Friends, myself Dipankar Majumdar. Welcome to my channel Microbiologist's Forum. Today I will discuss about the basics of pedigree analysis, and this lecture is divided into three parts. In the first part I will tell you what is the pedigree and what are the symbols used, how many types of traits, etc. And the next two lectures I will discuss about the analysis of different traits. Now, pedigree. What is pedigree? Pedigree is the representation of family history by using internationally recognized symbols. To any investigator dealing with the human genetics or clinicians, a person who is having a particular trait and which is interested to the investigators, and it is transmitted from one generation to another generation, then this person who is affected first, or the trait is reported first to which individual, is known as proband or index case. Or when the individual is a male, it is known as propositus, and when the individual is female, it is known as proposita. And modes of inheritance describe the different ways in which the genes handled down from parents to their offspring through several generations may express themselves. Genes exist in pairs on homologous chromosomes except on the X and Y. For female it is XX and for male it is XY. And pedigree analysis is done only in case of human beings. For other animals it is not done. And these are the internationally recognized symbols. Please see the chart so that you will understand, and I will discuss later about the special symbols. One is consanguineous mating, where mating is not properly defined. Another is parent with son and daughter, and carrier X-linked is denoted by a symbol that is also shown, dead individual is also shown, and twins, dizygotic twins, monozygotic twins, everything are shown, and sex unspecified is also shown. You have to practice the symbols and memorize it for the analysis of the pedigree. Now, what is the quantitative trait locus or QTL? It is a locus that correlates with variation of a quantitative trait such as SNP, that is single nucleotide polymorphism, and AFLPs, amplified length, fragment length polymorphism, etc. And what is a LUCA, last universal common ancestor or concestor? In July 2016, scientists reported identifying a set of 355 genes from the last universal common ancestor of all organisms on Earth. LUCA or concestor is the most recent population or organism from most recent common ancestor, that is MRCA, of all current life on Earth. Pleiotropy, in Greek pleion means more and tropos means way, that is pleiotropy occur when one gene influences two or more seemingly correlated phenotypic traits, such as gene that exhibit multiple phenotypic expression called pleiotropic gene. Example is PKU or phenylketonuria. Twins twins are mainly of two types, monozygotic twins, and that is developed from one zygote which splits and form two embryos, and these are identical twins. And dizygotic twins, each twin developed from a separate egg, and each egg is fertilized by own sperm, that is non-identical twins or fraternal twins. Female twins or sororal twins, male twins or fraternal twins, female male twins, most common pairing, that is 50%, or female male 25%, male female 25%, etc. And Siamese twins or conjoint twins, twins where share their vital organs, they dies before becoming adult. Now, the mode of inheritance, there are 1364 disorders where mode of inheritance have been firmly established. These include 736 autosomal dominant, 521 autosomal recessive, 107 X-linked disorders, and further 1447 disorders where unifactorial inheritance is suspected but not yet proven. And this is the X and Y chromosome. The left hand side chromosome is X chromosome and right hand side chromosome is the Y chromosome of human beings. And there is a catalog of human genetic traits to track of genetic disorders and the genes that control them. Victor McCusick, a famous human geneticist from Johns Hopkins University, and his colleagues have compiled a catalog of human genetic traits. The catalog is now published and available on World Wide Web, that is online inheritance, online Mendelian inheritance in man, that is OMIM. OMIM is the online version. The original version was the MIM that was compiled by McCusick and his co-workers in 1966. MIM, the full form of MIM is Mendelian inheritance in man, and there are 12 editions between 1966 to 1998. Nearly all of the 1486 entries in the first edition of MIM discussed phenotypes. Online inheritance, online Mendelian inheritance in man, OMIM, is a continuously updated catalog of human genes and disorders and traits with particular focus on the gene phenotype relationship. As 28th June 2019, approximately 9,000 of the over 25,000 entries in OMIM represented phenotypes. The rest represented genes, many of which were related to known phenotypes. And this is the cover page of the book written by Victor McCusick on human genetics. And this is the home page of OMIM, and it is the source, online source regarding the human disorders. That's all. Thanks for your patience watching. In the next coming two videos, I will discuss about the analysis of the various traits, and the traits are mainly of two types: autosomal traits, sex-linked traits. Third one is the trait controlled by mitochondria. Among autosomal traits, there are two traits, autosomal dominant and autosomal recessive. And in case of sex-linked inheritance, these are X-linked dominant, X-linked recessive, and Y-linked characters. Okay. Thank you. Thanks for your patience hearing.